So hi guys, in this video we are going to discuss about several cell cycle regulations, right? So let us see what are the topics of discussion. The first one is the how the S-cyclin CDK acts, what is CD6, and what is retinoblastoma or RB protein, right? So these are the three things that we are going to discuss in this video. So SCDK is basically S-cyclin and CDK. So before that, coming to S-cyclin and CDK, first we have to know about the CDC6. Okay, so it is a protein basically, and it actually prevents the DNA replication until all the checkpoints, right? All the checkpoints of G1 are met. Okay, if all of them are completed or satisfied, then only the cell cycle will proceed, right? It does so that it does not proceed with damaged DNA. That is why CDC6 is present. So what happens is there are certain structures known as the origin recognition complexes, right? And this is a blue colored line represents a double stranded DNA, and this is the origin recognition complex, which actually binds to sites in the DNA. Okay, binding sites are present for this origin recognition complex. So here it binds. And what is the origin basically? Origin is basically a place in the DNA from where the replication starts. Okay, we will study about that in the replication of DNA. In human DNA, there are multiple origin of replication, right? So this is, suppose this is again the double stranded DNA. This is the origin recognition complex, and what normally happens is this CDC6 actually binds to this origin recognition complex, right? And because it is bound to it, there is no replication going on. And this CDT is also another protein. So what happens, this CDC6 and CDT actually recruit the hexameric helicase known as MCM27. Okay, this MCM is basically the hexameric helicase. So what happens is, so this is the basically the structure, and CDC6 is still present. Now these are the hexameric units of MCM, and what happens is it gets organized at the sites, okay, like this, and this structure is known as the pre-replicative complex. Okay, so this is the pre-replicative complex and formed in the G1. Now suppose all the checkpoints of G1 are met. Okay, if all checkpoints are met, then the S-cyclin and CDK will form. So cyclin is activated only, the cyclin dependent kinases are activated only after the cyclin binds to it. Okay, so this complex forms, and this acts on the pre-replicative complex. Okay, pre-replicative complex, and the CDC6 is removed, phosphorylated and removed from the complex. Now the origin recognition complexes are free. Okay, and therefore what will happen is, this is the replication bubble or origin of replication. It opens the DNA basically at certain sites. So you can see now the enzymes can go or get between the DNA and replication can proceed. Okay, so this is only happen if all checkpoints are met, and DNA will then be replicated, right? We'll discuss the details of replication in the video about replication. So now what happens after replication is we get two strands of DNA, and it is semi-conservative because one of the parent strand will remain and another new strand will be synthesized. Okay, so hope this part is cleared. Now coming to the last important regulator, and that is the retinoblastoma protein. Okay, so what retinoblastoma does is it prevents the transcription of the genes for S phase proteins. Okay, so several proteins are needed for S phase. So retinoblastoma prevents the transcription of those proteins until the checkpoint is, all the condition of checkpoints are met. So suppose these are the genes, the violet color represents the genes, and this is a transcription factor. Okay, E2F is a transcription factor, and this is the retinoblastoma protein which is bound to the transcription factor, and therefore no gene transcription is going on right now. Okay, mRNA cannot be synthesized. This is the inactive. E2F is a transcription factor or inactive gene regulatory protein. Now what happens, S-cyclin CDK comes and it phosphorylates the retinoblastoma. This is again the gene that needs to be transcribed or translated, and this is the E2F, and this is retinoblastoma. So now see, once it is phosphorylated, the retinoblastoma detaches from the E2F, and therefore the gene transcription and translation can proceed. Okay, and the cell will move from G1 to S phase. So this is it. If you like this video, please subscribe to my channel. You can also follow my Instagram and Telegram for access to handwritten notes. And as always, thank you for watching.